virus or pathogen. In all organisms that carries the genetic code to the ribosome within the cytosol to synthesize new proteins within the body. mRNA-based vaccines are made up of a single-stranded mRNA, which uses the genetic code of a virus or pathogen to encode a disease-specific antigen of the virus, such as the SARS-CoV-2 spike protein. Thus, mRNA vaccines are non-infectious, as no viruses are used in the vaccine production, but instead use a DNA template, making production more rapid. Without the use of viral vectors or adjuvants in vaccine administration, the mRNA is introduced into the body and released within the cells. mRNA vaccines work in the cell's cytosol, not the nucleus, and thus do not integrate into the cell genome. The mRNA is translated and the encoded antigen is presented on the cell surfaces by antigen-presenting cells. Once produced within the body, the antigens elicit an immune response by generating both a T-cell and neutralizing antibody responses. Neutralizing antibodies neutralize the virus so it is no longer infectious, and CD4 positive helper T-cells trigger immune responses, including CD8 positive cytotoxic T-cells response to kill virus-infected cells, thereby teaching the immune system to recognize and attack the virus, and preparing it to recognize the virus when later exposed by forming memory T and B cells and preventing the disease. mRNA vaccines may be administered multiple times, and these boosts might help immunity should it decrease in the future. There are many different types of vaccines used to help prevent infections, all of which have the same goal: to train your immune system to recognize and defend against viruses or pathogens that cause disease. Vaccines have been a key development to protect against infectious disease, and novel advanced vaccine technologies have been introduced over time. Emerging mRNA-based vaccine technology is set to potentially revolutionize how we tackle emerging infectious diseases in pandemics, and has been studied by scientists for over 20 years. mRNA, or messenger RNA, is a single-stranded molecule found in all living organisms that carries the genetic code to the ribosome within the cytosol to synthesize new proteins within the body. mRNA-based vaccines are made up of a single-stranded mRNA, which uses the genetic code of a virus or pathogen to encode a disease-specific antigen of the virus, such as the SARS-CoV-2 spike protein. Thus, mRNA vaccines are non-infectious, as no viruses are used in the vaccine production, but instead use a DNA template, making production more rapid. Without the use of viral vectors or adjuvants in vaccine administration, the mRNA is introduced into the body and released within the cells. mRNA vaccines work in the cell's cytosol, not the nucleus, and thus do not integrate into the cell genome. The mRNA is translated and the encoded antigen is presented on the cell surfaces by antigen-presenting cells. Once produced within the body, the antigens elicit an immune response by generating both a T-cell and neutralizing antibody responses. Neutralizing antibodies neutralize the virus so it is no longer infectious, and CD4 positive helper T-cells trigger immune responses, including CD8 positive cytotoxic T-cells response to kill virus-infected cells, thereby teaching the immune system to recognize and attack the virus, and preparing it to recognize the virus when later exposed by forming memory T and B cells and preventing the disease. mRNA vaccines may be administered multiple times, and these boosts might help immunity should it decrease in the future. There are many different types of vaccines used to help prevent infections, all of which have the same goal: to train your immune system to recognize and defend against viruses or pathogens that cause disease.
Ladies and gentlemen, welcome to Biontech's digital press conference. Today, the leadership team will provide an update on Biontech's vaccine development program. You will see a presentation by Ugo Shahin, CEO and co-founder of Biontech, Özlem Türeci, Chief Medical Officer and co-founder, and Sean Merritt, Chief Business and Commercial Officer, and Dirk Potting, Chief Financial Officer. After the presentation, there will be a Q&A session, and you will be able to ask questions via the chat function. I will now hand over to Sean. Thank you.
Yeah, good morning, everyone, and welcome to our conference. Just to remind everyone, this slide presentation has forward-looking statements in it. And for those of you who have got up in the US to listen to this, very brave at 3 a.m. in the morning, just to remind you of the safety information related to our vaccine use in the United States. Okay, so we're here today really to give you an update, and of course one of the things that has recently occurred is conditional marketing authorization received yesterday from Europe in 27 European states for our vaccine, and this is together with Switzerland one of the first conditional marketing authorizations worldwide. Today we have use either through temporary use, emergency use, or now conditional marketing authorization in over 45 countries. And we'll see in a minute where our vaccine is being used currently. And today is the launch of our brand name, Comirnaty, which is the official name of the mRNA vaccine in the European Union and Switzerland. So just on the brand name itself, just to give you some understanding of why we picked this brand name, this was developed from over 1,000 potential names in April. We ended up with a short list of 3, and we picked Comirnaty, which really represents several things. Of course, it's a vaccine against COVID-19. It contains mRNA in the name, which is our key technology for the vaccine, but also how it works. The I stands for immunity, and it encompasses community. And that's something that we have really experienced as a company, just the coming together of not only with our partner Pfizer, but actually with academics, clinicians, and governments worldwide to address this pandemic. And it's probably, in my life, been one of the best collaborations across those groups I've ever seen. So I mentioned that just now. It's really a concerted and large-scale global effort. You see here on this map the dark green states where we have the conditional marketing authorization. We also, in the lighter green, approved emergency use authorization in those countries. And then in the lightest green, this is where our vaccine is currently being used in North America, the United Kingdom, and Israel, of course, which is not represented on there, but of course is being used. Our goal from the beginning was always to commit to an equitable supply of vaccine globally. It doesn't matter which country you are. If you want vaccine from us, we will supply it. We supply it through supply contracts. This year, we have a target of 50 million doses by the end of 2020, so by the end of next week. And being supplied to other countries, Europe now and North America. With respect to Europe, how have we in the European Union decided on who gets how many doses? This was decided between the European Commission and its member states, and is based on the member states' populations as a principle. All countries across the EU that have requested doses will receive them in the next 5 days, the very initial supply, and that will be followed up next week with further supplies. We are just completing actually the paperwork now, which is to just check the quality once more on what we've produced before releasing it for packing and shipping, and that's occurring as we speak. Just on transportation, just to remind everyone, we transport in what are called freezer boxes or shippers. These have been designed together with our partner Pfizer to hold between 1,000 and 5,000 doses per box. They are shipped in dry ice to keep them at -70. They can be put in planes, they can be put in lorries, on boats, and they have a GPS tracker to ensure that we can monitor on a minute-by-minute basis the temperature in the boxes. And these are delivered to vaccination centers where they can be either left in the shipper for up to 15 days if you re-ice, or in regular fridges. And this slide here emphasizes that a bit more. Just to remind everyone, the administration is to vaccinees is at room temperature. We've seen the pictures on the television now, people rolling up their arms and getting the vaccine, intramuscular injection. Once removed from the box, the unopened vaccine can be stored in the fridge up to 5 days at 2 to 8 degrees, or if you take it out and put it on the bench at room temperature, up to 30 degrees, 2 hours prior to use. As I mentioned earlier, these shippers can be used as -70 freezers in their own right. You can open them twice a day, once in the morning, once in the afternoon to take vaccine out, and you can re-ice them every 5 days for up to 15 days, and there you have a -70 temporary freezer. But if you want to store long term, and some countries are doing that, you can store in a -70 freezer for up to 6 months. Just as an example, I mean, Biontech is responsible for distribution in several countries, Germany being one of them, our home country. And here, this is an example of the distribution chain. We will deliver to 25 distribution centers, which are run by the federal states within Germany, who will then further distribute to just under 300 districts, 450 vaccination centers, and includes 100 mobile vaccination locations to get to those those areas where there are less people and less population. This is all supported by online information. There's a lot of technical information for doctors, nurses, pharmacists, people who are handling the vaccine, and that's complemented too with paper copies on the medical information and the use of the vaccine and how to administer it. We hold webinars, and yesterday we held a webinar, and just under 2,000 people registered for that across Germany. This was a professional for the professional people administering the vaccine, and we had over 1,100 questions which we were able to answer. So there's a lot of support from Biontech. Pfizer, our colleagues in Pfizer are doing similar programs across the world too, and Fosun will be in China doing the same once we launch in China. So I think that's just a little bit of an update on the distribution, and then I would like to hand over to Özlem for the next part of our presentation.
Yes, thank you very much, Sean. So I'll start us off by leading you through the last 11 months, which for us were a journey of developing a vaccine at light speed while adhering to the highest standards of science and of ethics. We started shortly after the genetic sequence of SARS-CoV-2 was published in January. mRNA is a very versatile technology, which meant that we could embark in a true vaccine discovery and rational selection process, starting off with more than 20 vaccine candidates. By March, we had selected, based on preclinical studies, the 4 best among them, and embarked into partnerships with 2 renowned partners, with Pfizer and with Fosun. We decided to go into these partnerships because we knew that developing, manufacturing and also distributing a vaccine worldwide would need global joint efforts. In April, we started our clinical testing program with 4 of our candidates, comparing them, and in June we were at a stage where we had narrowed down to 2 candidates, and eventually BNT162b2, the best of best among our candidates, was selected for the pivotal trial, which started in July, which was a global trial to assess efficacy and safety of our vaccine. In November, we could present the final data of our efficacy and safety analysis for our primary endpoint, and had already started in October rolling submissions of the data we were producing to regulatory authorities, which allowed us to file pretty quickly those dossiers for emergency and conditional approvals, which Sean has just mentioned. And we are now in the midst of a global rollout. Let me explain very briefly how our phase 3 trial was set up. This was a randomized placebo-controlled trial in approximately 44,000 participants, most of them at the age of 18 to 85. These participants were healthy, which included participants with chronic diseases such as cardiopulmonary disease, diabetes, etc., as long as these were stable. The participants were treated either with placebo or with our vaccine, BNT162b2, administered intramuscularly in 2 injections, 21 days apart. And these participants were were under active surveillance, triggering telehealth or in-person assessments when COVID-19 symptoms occurred. And for efficacy, we counted in both arms the number of confirmed COVID-19 disease cases starting 7 days after the second dose. And this all was under the supervision of an independent data safety board. Counting of cases in both arms provided us with the split of cases, disease cases you can see here, while we had 162 cases of COVID-19 in our placebo arm. The vaccine arm of BNT162b2 immunized individuals only showed 8 cases. And here you have a bit more data showing you what is called Kaplan-Meier curves on the right side of this slide, which is the cumulative plotting of symptomatic COVID-19 cases. The white curve is those patients with COVID-19 which occurred in the placebo arm over time, and the green one is our vaccine arm. What you can see is that both curves clearly diverge. What is also important to note is that this divergence occurs early, already in between those 2 applications of the 2 vaccine doses, so that the onset of protection appears to begin as early as 12 days after the first dose. With this case split 8 to 162, we have a vaccine efficacy of 95% after 2 doses were given, starting 7 days after the second dose. We have also assessed the vaccine efficacy in different subgroups which were treated and vaccinated in this trial. And here you see the case split, meaning the disease cases occurring in the vaccine arm on the left and the placebo arm on the right, in subgroups with different, what we call comorbidities, meaning underlying chronic diseases, including cardiovascular disease, chronic pulmonary disease, diabetes, obesity, hypertension, and others. And as you can see also here, the vaccine efficacy is consistent, in particular in those groups in which we have sufficient cases to assess. We have also looked into other subgroups and can say that we have this high vaccine efficacy, around 95%, in across ages, across genders, across ethnicities, and as I have also shown to you, across comorbidities, including in subjects of 65 years of age and older. The Independent Data Monitoring Board has not reported any serious safety concerns in our study. The vaccine is generally well tolerated. The observed side effects in our study were common reactions to vaccination, and these were transient. The adverse events were usually mild to moderate in intensity and resolved within a few days of vaccination. Most frequently, we observed adverse events such as injection site pain, fatigue, headache, muscle pain, joint pain. With regard to grade 3 adverse events, greater than in 2% of vaccinees, we observed two, namely headache in 2% of vaccinees and fatigue in 3.8% of the vaccinees. As you have already heard from Sean, the vaccine is being rolled out in various countries so that in the meantime, thousands of individuals have been vaccinated. In these rollouts, the safety is being monitored so that we will be able to also assess safety in these very large populations. And in addition, we are continuing to observe the vaccinees in our trial for up to 2 years after receiving the second dose. And with this, I would hand over to Ugur.
Yeah, thank you, Sean. Thank you, Özlem. I would like to give. I would like to give a short introduction about what is messenger RNA. It's a completely new drug class, how it works, and how the vaccine works. And I would also like to explain you how we deal with, for example, a changing virus, with new virus variants upcoming, and what it does mean for the vaccine activity. And so on this slide, you can see the most important biological principle in nature. We have in our every cell in our body has DNA, which is hard-coded information. You can compare that with information, for example, on your computer on the hard disk. We have in our cells messenger RNA, which is the working information, which provides instructions to make proteins. And we have proteins, and the way how this is managed in biology is DNA information is used to make messenger RNA information, working information, and this is used to make proteins. And what is really important is that messenger RNA is the first molecule in life, so it's existing for hundreds of millions of years. It is involved in all aspects of our cellular biology. And what we do when we make messenger RNA vaccines, we make a copy of a messenger RNA which looks and resembles the natural mRNA molecule. So that means we use only natural components which are existing, and thereby we can rely on the biology of messenger RNAs, and that this is a temporary information, this is a temporary message, and it is deleted and degraded, fully degraded after a few days. And this is in principle the revolution, that we can communicate with our cells, providing temporary information, and after the instructions have been fulfilled, the messenger RNA disappears, and this brings in a safety by design. So how did we use that? How did we use that for making a COVID-19 vaccine? So as already mentioned, this is a molecule, it's a biological molecule. It is known for over 50 years, and it is researched for over 50 years. Actually, we know much more about messenger RNA as compared, for example, to other types of vaccines, because this is a biological molecule which is studied for decades by basic scientists as well as by scientists using messenger RNA for making vaccines. And how does it work? So we take a piece of information of the virus, which is also consisting of RNA, and we take only a piece of information which is not able to form a virus, but to form a part of the virus. And the most important part of the virus is the so-called spike protein, which helps the virus to enter into human cells. And we put this information into our messenger RNA vector, which has additional information to ensure that the messenger RNA, when it comes into cells, is recognized, is used to make the protein. The messenger RNA itself is not stable. As I said, it is degraded very fast, and to ensure that it is not completely degraded before it reaches the cells, we encapsulate the messenger RNA into lipids. This lipid components protect the messenger RNA and also ensure that the messenger RNA is transported to the right cells, to a certain type of immune cells. And this lipid nanoparticles can be solved in buffered solution, in aqueous solution, and then used for intramuscular injection. It can be produced very fast in clinical grade, and since we do that in a synthetic manner, we don't need to make messenger RNA vaccine in cells. So we can make the vaccine just in the laboratory without using any animal material. And what is really fantastic about this technology is that we have in our vaccine only the genetic information of this piece from the virus, but nothing else. So that allows us to communicate with the immune system in an extremely precise fashion. The production is done in the laboratories, and the production can be scaled up from amounts for a few thousand people to amounts to many millions, hundreds of millions doses. And we did that exactly in our project by installing manufacturing capacity, which could allow us to make more than 1 billion doses. So immunologically, what happens is a vaccine is nothing else as a tool to inform the immune system about a piece of the virus, so that the immune system can see this piece and is prepared and able to recognize later on when the vaccinated person is encountering the real virus. How this works in the immunological manner is that the lipid nanoparticle, which carries the mRNA, is taken up by specialized immune cells, and the specialized immune cells make this virus protein, the spike protein, and the spike protein is recognized by two important cell types. These are T cells, which help to recognize virus components even if they are hidden in the cell, inside of the cell. And we have another immune response. This is the antibody response. And if the antibody response is functional, then these antibodies could inactivate the virus. So that means these antibodies could help to prevent that the virus enters the human cells. And we need both type of immune responses. The antibodies, which could prevent the virus from uptake, as well as the T cells, which are able to deal with the virus when some virus particles have managed to enter into the cell, so that the T cells could fight and eliminate the cell. What is important is that messenger RNA is a transient information. It is gone after a few days, but the immune system has a memory function. So that means the information which was provided by the messenger RNA could be memorized in principle by the immune system, and particularly by T cells, for several years. What we don't know at the moment, how long the memory immune response that we induce against this virus lasts. We have at the moment data supporting a memory immune response for 3 months, and we are collecting further information for evaluating immune responses up to 2 years. So during the development time of 11 months, we not only did clinical trials, but we did a number of mechanistic studies. We investigated how this messenger RNA works in preclinical models. These are all information resulting in publications. In total, we have now 5 publications explaining how this messenger RNA vaccine works, which type of immune responses are generated, how it is tolerated, and how is the clinical efficacy in the groups as them shared. What is important is that the immune response that we are getting against this virus is directed against multiple components of this spike protein, and this is something which is really important. I will show you why this is important. So on the left side, you see the model of the virus, and you can see the red structures, which are the spike proteins. And as you can see, the virus has multiple spike proteins, and the depiction of the spike protein, this is a 3D modeling of the spike protein. It consists of small pieces which are called amino acids. And these amino acids, in total, the spike protein has more than 1,270 pieces, amino acids. And these pieces are encoded by our messenger RNA vaccines. That means that we have used the full protein of the spike protein to make the vaccine. So we know that this virus is not stable. No virus is stable, and this virus evolves. It is an evolution where the virus gets more fitness. But these mutations in this coronavirus are most often singular mutations or several singular mutations. And what we did in the last months, every time when a dominant mutation, a virus with a dominant mutation came up, we evaluated whether the vaccine response induced by our vaccine, the antibody response, is able also to prevent the infection by the new virus. And as you can see on the right side, there are a number of variants, and you see numbers, for example, Q321L. So this is a mutation at the position of 321, where one amino acid changed to another amino acid. So and you can see that the neutralization, the activity, these are the bars against all these variants are very similar. So that means even though our vaccine contains only the wild type, these mutations are recognized. So, and we have now a new variant, the variant which was discovered in UK. And this variant is a little bit different. It has 9 mutations, not only a single mutations. And we don't know at the moment if our vaccine is also able to provide protection against this new variant. But scientifically, it is highly likely that the immune response by this vaccine also can deal with the new virus variants, because, as I said, the vaccine contains more than 1,270 amino acids, and only 9 of them are changed. So that means 99% of the protein is still the same. And we know that the antibody response and the T-cell response against this spike protein that we induce with this vaccine is polyspecific. That means multiple sites of the virus proteins are attacked, and therefore we have scientific confidence that the vaccine might protect, but we will know it only if the experiment is done, and we will need about 2 weeks from now to get the data, and will of course publish the data as soon as possible. So a few words about the components in the vaccine. I shared the information that the messenger RNA is in the vaccine. We have in the vaccine the lipids. Yeah, one of the lipid components has also a component which is called PEG, which helps that the lipids or the lipid nanoparticles remain stable. We have in our vaccine buffer solution salts, 4 different salts, and we have sugar, which allows us to freeze and thaw the vaccine without losing stability. So these are the chemical components of the vaccine. The messenger RNA, the sugar is sucrose, the lipids have complicated formulas. Most important information is that one of the lipids is pegylated, and then we have salts which have been also used in other type of pharmaceuticals. So this was my last slide, and we are happy to take your questions.
Dear ladies and gentlemen, we are now starting the Q&A session, and please ask your questions via the chat function. So the first question is also about the mutation. If a significant mutation of the COVID-19 virus were to arise tomorrow, how rapidly do you believe that this vaccine could be developed?
So in principle, I will take this question. In principle, the beauty of the messenger RNA technology is that we can directly start to engineer a vaccine which completely mimics this new mutation, and we could be able to provide a new vaccine technically within 6 weeks. So that means a vaccine which contains this information. Of course, this is not only a technical question. We have to deal with the question how regulators, how EMA, FDA, and other authorities would see that. But in principle, technically, it is possible to make a new vaccine mimicking the new strains within a few weeks.
And also a follow-up question from Handelsblatt regarding the virus mutation in Great Britain. What makes you optimistic that your vaccine will be effective with this mutated virus too?
Yeah, so as a scientist, you are not optimistic, but you think in likelihoods. So the likelihood that our vaccine work, as I said, is relatively high, because even though there are multiple mutations, there are much more sites which are not mutated. We had already a look into, for example, the sites in the virus which are recognized by T cells, and most of the sites which are recognized by T cell responses are still conserved in this new virus variant. So that makes us confident that the T cell response works, and multiple antibody binding regions are also conserved. But as already mentioned, we need to do the experiments to quantify how well our vaccine works against this new variant.
The next question is about manufacturing. Where in Europe is Comirnaty manufactured? When will the Marburg site begin manufacturing?
I can take this. Comirnaty is at the moment produced at various sites. So, for example, in Mainz, at a site in Halle, Derma Farm, at Polymoon, in close to Vienna, actually, which we mentioned before. It is also produced partially and filled and finished at the moment in Puurs in Belgium at the Pfizer site. So we have a complex network already of various sites in Europe, plus we have Pfizer's network in the US. So The Marburg site itself, there's a full tech transfer ongoing right now. So we signed and closed the deal with Novartis. It's our site now, and we are anticipating to run first production batches in February, actually. So the whole process is ongoing, and February we'll start producing in Marburg, with a release probably in end of Q1.
The next question is about BioNTech's strategy. Will and if how this change the strategic focus of BioNTech? In detail, more focus on vaccine development and less on cancer treatment?
You want to take that?
You take that. You take that, Hugo.
Yeah. So this is this success of our infectious disease vaccine shows that messenger RNA therapeutics or vaccines have really groundbreaking potential, allowing us to make new vaccines and other types of molecules in a short time. We have at the moment multiple messenger RNA therapeutics in clinical testing for oncology. We have already infectious disease pipeline, including an influenza vaccine project co-developed with our partner Pfizer. We have a vaccine project to develop vaccines against tuberculosis and against HIV, and we will certainly consider to start other infectious disease vaccine projects. But of course, our projects in oncology, not only the messenger RNA-based vaccine projects, we have gene therapeutics, we have monoclonal antibodies, including bispecific molecules, we have a small molecule in clinical testing. We will continue that, and the success will allow us to further accelerate that. Our aim is to bring personalized medicines also for treatment in cancer to the market, to address medical need for individual patients.
The next question is from CBN. Dr. Sahin said last time that BioNTech will study if the vaccine will prevent people from infection. How's the studying going on?
So we are collecting data, and we expect that end of January or in February we will be able to update and provide information whether the vaccine is able to prevent asymptomatic infection.
So the next question is from Südwestrundfunk. Why hasn't the vaccination started yet?
I'll take that one. It has actually globally. I mean, we've started vaccinations in the United States, in Canada, in Israel, in the United Kingdom. In Europe, we received approval yesterday, and as I mentioned, we're just finishing the paperwork now to distribute throughout Europe, all 27 member states, such that everyone can begin vaccination at the same time, and that's going on at the moment to ensure that we can start vaccinating really very, very quickly.
And where is the vaccine for Germany produced?
So as I mentioned before, we have these various sites and partners, collaborators on this process. So currently, all of the product that will be shipped to Germany and Europe is in pours, and it was filled and finished in pours. The components came actually from different parts, a portion from the US, a portion from Germany, from our sites and our partners. So it's filled and finished in pours, and it's all sitting in pours right now. And we have a very tight delivery schedule into all the European countries, so that there will be a delivery into every European country, into every EU country on the 26th. So every country can start on the 27th with the vaccination.
And do you plan to open other BioNTech locations in Germany?
Yeah, for manufacturing, we are scouting every possibility right now to produce as many vaccines as possible in 2021 and then thereafter. So at the moment there are various plans, but they're not very specific yet. But we are working on plans actually to expand the production network as much as possible.
The next question is from Wall Street Journal. Does the new mutation or a potential next mutation change the calculation of what portion of the population should be vaccinated to achieve herd immunity?
Oh, this is an excellent question. We don't know. So the topic of herd immunity, there are always this discussion about 60 to 70%. But if the virus becomes more efficient in infecting people, we might need even a higher vaccination rate to ensure that that normal life can continue without interruption. But 60 to 70% of vaccination rate will really dramatically change the situation. It is not that we have to deal with outbreaks if the virus becomes more effective, but it still would provide a normal, more or less normal life.
The next question is from the Financial Times. Are you seeing or do you anticipate any UK supply issues because of travel chaos and Brexit?
No, we do not. As I said last time, there are multiple routes into the UK, not just through the Channel Tunnel.
And then the next question is about the vaccine production. What steps of the vaccine production process are done by BioNTech itself? Who are the partners for the other steps?
So as I mentioned before, so there is a complex network. So various pieces are done at various steps. So for example, drug substance, the first step is done in Mainz. The mRNA molecule is produced, and our partner Rentschler actually does a certain purification step. There is drug product formulation, which is then putting the lipids together with the mRNA at our site in Dermafarm, at our partner site, Dermafarm and Polymoon, as I mentioned before. Pfizer itself in Puurs is doing that as well. So they are producing drug product as well. So they're putting together the mRNA and the lipids. Fill and finish is for Europe is currently solely done at the Puurs site at Pfizer. But we have a network coming online actually of various suppliers for fill and finish actually as well. So as I mentioned, there are various parts and bits and pieces. So it's a complex network, but there will be one vaccine in the end.
So the next question is from Asahi TV from Japan. The vaccine has been submitted to Japanese regulators. How is it going? Are you in contact with them, and how is this conversation going on?
Yes, there has been a bridging study in Japan, which is ongoing, and the data of the Japanese study, as well as the data of the global program, are being discussed with the Japanese authorities, who, as also other authorities, are very diligently assessing the data. And I cannot give any timelines now, but would expect that the process in Japan is also proceeding very fast.
And are you confident it's safe for Japanese people? How much data do you have from trial participants of Asian origin?
Approximately 4% of our vaccinees in the global trial were of Asian origin, and as I already said, we have also a dedicated trial in Japan ongoing.
The next question is from Berliner Zeitung. Are you testing the vaccine in pregnant women and children as well?
Yes, there is a dedicated, broader clinical development program with several studies planned for next year, which will look into defined populations or subgroups. This includes a study which will vaccinate pregnant women, a study which will assess children of younger ages, and also a study with regard to immune-compromised individuals. These programs are being actively discussed and planned together with regulatory agencies.
And can the interval between the two vaccination shots be shorter or longer than 3 weeks?
We recommend it not to be shorter, but a longer interval is allowed, meaning 21 days and longer.
And the next question is from Politico. When will the first shipments of the vaccine will leave warehouses in the EU?
So that could be as early as tomorrow.
And how many doses BioNTech expects to have for EU countries by the end of 2020?
12.5 million doses.
And can you give us your assessment of the new virus variant that emerged in the UK and what impact it might have on the BioNTech Pfizer vaccine? Do you need to adapt the vaccine?
So I shared already the information at the moment. We don't see a reason for adaptation of the vaccine, but we have to wait for results which will come in 2 weeks.
And there's another follow-up question. You said you would need 2 weeks to test the vaccine against the UK variant. Can you elaborate a little bit more? Do you have the virus at hand, or are you engineering the mutations into a wild-type virus?
Yeah, so that what we do is we know the mutations, and we engineer the variant by mutating, indeed doing the mutations in the wild type virus, and then we take this component, this mutated component, and bring that in our assays, and then we can compare in these assays how well the immune response generated against the wild type virus component is able to inactivate so-called pseudoviruses. These are not real viruses, but they behave like real viruses, so that we can work with them in normal laboratory conditions, and this will give us a number. Yeah, if the mutant virus is inactivated in the same manner as the wild type virus, we would say there is no difference. If we see a reduction, we could quantify that, and this could give us also an idea if it makes sense to consider to make a new vaccine. But this is really hypothetical at the moment. As already said, we believe that there is no reason to be concerned or worried until we get the data. Yeah.
And the next question is from SWR. When will you have data to clarify whether the vaccine also prevents spreading the disease?
We can repeat what you already said, Ugur. These assessments based on data we have produced in our global trial are ongoing, which also involves testing the samples of vaccinees we have immunized. So the technical assays are sort of the bottleneck, and we expect to have the data sometime latest in February.
Yeah. So it is so the type of studies which are done for evaluating the secondary spreading are not suitable. But what we will learn from this from the study that Özlem said discussed in February is whether we prevent asymptomatic infection, and this is highly correlated with the ability to prevent transmission from an infected person to another person.
And what will be the total 2021 capacity? Are capacity extensions planned?
Yeah, so the total by end of 2021 will be 1.3 billion doses. This is the entire network Pfizer combined with BioNTech right now. As I mentioned earlier, because we are the only EU vaccine right now with the potential further authorizations coming soon, but there will be scarcity of the vaccine, and we try to optimize our production processes, and we are looking into expansion of the network even further.
The next question is from the Wall Street Journal. Just to clarify, did you say it will take 6 weeks to develop a vaccine against the UK mutation if your current vaccine is not effective?
I said technically we could come up with a new vaccine batch in 6 weeks. But this is really a technical consideration. Since this new vaccine would not have efficacy and safety data, there would be a lot of discussion whether this new variant, the vaccine with the new variant, could be applied or not applied. So this is a more complex discussion. But theoretically, yes, in principle, a new mRNA vaccine with addressing this new variant could be prepared in 6 weeks.
If I may add to that, in principle, what we would do is to change the insert and replace a virus variant, the one we are using now, with a different one without touching the platform as such. And as Ugo has pointed out, it will depend on the extent regulators would accept the platform data we have produced with the current variant as being indicative for the new insert, basically into this platform. That will define how long it would take to approve that variant vaccine.
And how long are people immune if they are vaccinated today?
We do not know yet, and in order, we need basically the passage of time and the follow-up of the vaccinees to answer this question. As Ugo has pointed out, we currently, from our ongoing studies, have data for 3 months post the second dose of the vaccine. We see stability of the antibody response in particular, and we will continue to follow up. And according to what we see in a data-driven way, we will decide whether boosters of our vaccine are needed and in which time intervals. The good news is that mRNA as a platform allows frequent boosting, so that the further decisions will be really data-driven.
The next question is from Focus. Can you comment on your progress with formulating a vaccine that does not need to be stored in such extreme temperatures?
We will update on this end of January. The work is ongoing, and as communicated a few weeks ago, we are confident that we could come up with more relaxed conditions with regard to storage and with regard to stability at 2 to 8 degrees. But still, we are waiting for the results, and we'll update here end of January, beginning February.
The next question is from the Financial Times. How many doses do you anticipate you will be able to deliver to the UK by the end of the year, and how many by the end of January?
Yeah, I think that one is actually for Pfizer to answer, as it's considered to be a Pfizer territory. So there's been no comment on that from us.
And the next question: when life will return to normal, according to Professor Sahin?
Well, this is. We need a new definition of normal, and so the virus will stay with us also for the next 10 years, and we need to get used that there will be infections and small outbreaks again and again. But normal in the sense of that we don't need to go into a shutdown, that normal business life can continue, that we can reduce hospitalizations and deaths. That could happen. That could really happen by vaccination end of summer. And to be successful, of course, this not only depends on our vaccine. It depends really on the availability of other vaccines. So the vaccines of the colleagues from Moderna has now been authorized in the United States, and we are optimistic that there are other vaccines may be approved in the first half of 2021, and all these vaccine doses will add up to provide sufficient doses so that 60, 70, maybe 80% of the population can be vaccinated before autumn 2021. And this is really important because this winter we will not have an impact on the infection numbers, but we must have an impact and ensure that the next winter could be quasi-normal, the new normal.
We have then one last question from the Wall Street Journal. How many doses will have the US received by the end of 2020? And how many doses would the US and the EU receive separately each month after that?
Yeah, so as we presented earlier, there are 45 million doses to be distributed by the end of the year. We stated that in Europe it would be in this last week 12.5 million that we would be distributing. The United States approved the vaccine through emergency use earlier, and the number of doses will be around 20 million that will be delivered to the United States.
Thank you very much. We are now closing the Q&A session, and thank you for your attention.